Welcome to our channel Bio by Gulab Sir for Wisdom Coaching Center. As a part of series of lectures, today's topic is double fertilization. Double fertilization. Now see, what is double fertilization? To have the best understanding, before that, stigma, style and ovary. Here, as usual, you know that ovule with egg cells, 2 synergids, 3 antipodal cells and 2 polar nuclei. This is the developed female gametophyte, that what we learn. So to label, these are antipodal cells, polar nuclei or secondary nucleus. This is egg cell and these are synergids. Now, this is ready for fertilization. When pollen grain fall on the stigma, these fall in 2 stages, 2 cell stage and 3 cell stage. In the condition of 2 cell stage, that pollen tube developed, and when pollen tube begins to develop, that one generative cell divide into 2 and form 2 male nuclei. Now this pollen tube is continue to grow up to micropylar end. So just imagine this pollen tube is continue to grow up to micropylar. If it is said in 3 cell stage, that 2 nuclei which are present in generative cell that continue to transfer along with pollen tube. But in 2 cell stage, while developing pollen tube, that male gamete divide into 2, that you please remember. Now these pollen tube when transfer across its pathway and reach to the micropylar end, carrying these 2 male nuclei, these are suppose 2 male nuclei, and egg cell and secondary nucleus, that is polar nuclei, are ready to have the fertilization. That means this is first male gamete and this is second male gamete. So fusion of that male gamete, with egg cell, is known as first fertilization and form zygote. Fusion of this egg cell polar nuclei with second male gamete, leads to produce endosperm mother cell, also known as primary endosperm cell nucleus, So here this is very important, that this is diploid, male gamete is haploid and endosperm mother cell will be triploid. So these 2 fertilization together, fertilization 1 and fertilization 2, this together is called double fertilization. Where this one male gamete fuse with egg cell and another male gamete fuse with polar nuclei to form zygote and endosperm mother cell together is called double fertilization. This is called pre-fertilization. This is called pre-fertilization, and formation of endosperm zygote is fertilization. Now, what are the post-fertilization changes? As we know that here every plant, flowering plant, they have seed with testa and tegmen. Suppose inner layer called tegmen and outer one is known as testa. Yes, and this is inside the fruit. Now see what form that zygote develop into embryo. Zygote develop into embryo, and this embryo is that part present inside the seed with embryo axis. Endosperm mother cell either continue to remain endosperm or can be developed into cotyledon of the seed. And this is a ovary which present here. This is a ovary. This ovary after fertilization change into fruit. After fertilization change into fruit. So see, these are the pre-fertilization. That ovary is a pre-fertilization. Post-fertilization it become fruit. Egg cell with male gamete is a pre-fertilization. After fertilization, that zygote develop into the embryo of the seed. And this testa and tegmen, these are inner integument, later it form tegmen, that is inner layer of seed coat, this one, and this outer integument after fertilization form testa. This is outer covering of the seed. So here we learn that what are the parts of flower before fertilization that change into part after fertilization. So here you can make the part into two, pre-fertilization and post-fertilization. So here we can write pre-fertilization, the parts of the flower before fertilization and parts of the flower that modified into different organs or parts of the fruit after post-fertilization. So here double fertilization is completed. To understand these three cells together, to synthesis an egg is called egg apparatus, but here there is a finger-like cytoplasmic extension in synthesis is called filiform apparatus. Filiform apparatus that help to guide pollen tube to enter into the synthesis. Once again, what is filiform apparatus? Synthesis with finger-like projection, cytoplasmic, cellulosic finger-like projection. These are cellulosic finger-like projections which guide pollen tube in the synthesis for further delivery of the male gametes for double fertilization. Once again, double fertilization is the two-time fertilization, that is one male gamete fused with polar nuclei to form endosperm mother cell, another male gamete fused with egg cell to form the zygote. That zygote further develop into embryo and endosperm mother cell either remain endosperm in monocot, that is called endospermic seed, or is used to develop cotyledon with development of embryo, that is in dicot seed, and ovary that turn into fruit. Outer and inner integument of that ovule becomes seed. So outer part of the ovule, that is outer integument, develop into testa, and inner integument develop into tegmen, and thus here the double fertilization is completed, and that flower entirely being reproductive organ change into fruit with seed as to have reproduction, that seed once again germinate and new plant developed. I hope you did understand what is double fertilization. Question can be asked in board. What is double fertilization? Explain it with label diagram, or in NEET question can be asked ploidy. If a hexaploid male plant cross with a tetraploid female plant, what will be the ploidy of endosperm mother cell? or vice versa. This kind of question can be asked. Even if open book exam, if it starts, the question which will be asked on thinking basis. So what they will ask? Again, I repeat that question. If a hexaploid female plant cross with a tetraploid male plant. So if it is a hexaploid female plant, what would be the ploidy of egg cell? 3N. And what will be the ploidy of this polar nuclei? 6N. And if tetraploid male plant, what will be the ploidy of male gamete? 2N. So 2N with 6N, it will be 8N. So octaploid. Who? That endosperm mother cell. And what will be the ploidy of embryo, zygote? Naturally, if it is female plant was hexaploid, then egg cell will be 3N because meiosis, half number of chromosome will come there. And if male plant was tetraploid, that pollen grain male gamete will be 2N. So 3N plus 2N, it will be pentaploid. So ploidy of zygote is 5N and ploidy of endosperm mother is 8N. So this type of question, thought level question, will be asked on open book exam. And yes, it will be very, very interesting. Those who have learned conceptually, those who have clear concept, they will have ability to understand the question and apply the knowledge. Therefore, I appeal you to see my videos based on all the syllabus, so as to you find very easy to appear in open book exam. I hope you did understand. Even if you are in doubt, you can ask in comment section, or you can call me on mobile number 9898431081, as it is mentioned already in YouTube channel, so you can contact anytime. Also, I appeal you to like our channel and subscribe it and press bell icon to receive new videos as and when they are uploaded. Thank you.